Today is April 27th, 2014, and I am in the Senior Physiologist Lounge at the San Diego Convention Center, preparing to interview a long-standing member of the American Physiological Society, Dr. Joe Szurszewski. Joe has been a member of the American Physiological Society since 1973, and he's been affiliated with Mayo Medical College since 1971. Joe's research expertise is in the area of smooth muscle physiology, motility, and the control of the peripheral autonomic nervous system. And he's done stellar work helping us understand the role of smooth muscle in health and disease. Joe, welcome to the Society's Living History program.
Marty, thank you for having me. Being asked questions and giving me the opportunity to go over my career with you is especially nice. So thanks for fitting me into your schedule.
Well, it's a pleasure having you here, Joe, and I think not only am I interested in talking to you about your career, but I think there are a lot of fellow physiologists who are interested in understanding your career and how perhaps it mirrors their own career. So let's start by having you tell us a little bit about your family upbringing and how you got interested in science.
Right. So I grew up in the central area of the city of Pittsburgh, in an area that I would say might have been impoverished. Certainly was not an area of the city that was filled with resources. And I was told as a young child to get an education, and that was just reamed into my head almost on a weekly basis by my parents and my dad, who was, as they called him in those days, a laborer. I made it through grade school, which was important because most of the members of my family, when they finished grade school, did not go to high school. But I did get into high school, Central Catholic High School, which, and I look back at my life, was really a critical juncture because that high school taught me how to study. And they prided themselves on the fact that over 90% of their graduates went on to college. So getting into that school, and that was not a straightforward process, the diocese had to approve it because they funded those positions in the high school. So I got into that high school and was turned on to science by a physics teacher, and it was near the end of my high school time when Sputnik went up into the air. And so that created a lot of concern in the country, and so science suddenly was a focus for our government. From there, I went to Duquesne University, and I did so because that was the only one I could afford. And I remember vividly that the cost was $25 per credit. Now that seems like an awful small amount, but in those days, $25 was important, significant cost. But it wasn't the University of Pittsburgh or others. And it was at the university, Duquesne University, where I met a fellow who really pointed me in the direction of physiology, and that was Julius Greenstein. I'll never forget him. Cecilia and I, my wife, visited him a number of times in the last few years. But it was he who said to me, "You really ought to be a physiologist." This was kind of in contrary to the chair of the department, who said I should be a botanist, and that if I didn't submit my credentials to the University of Miami to be a botanist, she could not write me a good letter of reference. And so I took the chance. Drove out to University of Illinois, drove out to Michigan, University of Michigan, got back, decided Illinois was the one I wanted to go to and model my life after Julius Greenstein.
Well, I know you went to the University of Illinois, which was also my alma mater.
Yes, that's right.
And your doctoral advisor was my undergraduate advisor,
yes,
Frank Stegerda,
Stegerda,
who we euphemistically called the Rear Admiral, or the
That's right.
Gas man.
That's right.
Tell us a little bit about Frank Stegerda and the research you did at Illinois and the experiences you had with what was really outstanding faculty.
Well, it was an outstanding faculty. Dr. Stegerda, or when he wasn't around, we'd refer to him as Tech Stegerda. He was the student advisor at that time when I got there, so he took me under his wing, so to speak, and said, these are the things you need to do. He cautioned me about the physiology laboratory because at Duquesne University we used smoke drums to record our data, whereas at Illinois we had all the fancy grass electrical equipment. And he said, you know, you really need to study the manuals before you do research in the lab. So he was always right there trying to figure out what was...
Before you go on, you know, there's probably people listening today who aren't going to know what you mean by a smoke drum.
Oh my goodness. Well, this predated the ability to record with a ink pen, writing on a piece of paper coming out of a machine. A smoke drum is, if you think about it, is exactly what the words say. It was a drum shaped. You take a piece of paper and you smoke it. You put it in smoke generated by benzene. The paper became coated black. You wrapped it around a drum. You got a metal stylus, which was pressed against the paper. And that stylus in turn was connected to, let's say, a muscle fiber. And when a muscle fiber would twitch, the pen would go up, the drum was rotating. And so now one had a smoke drum record of a muscle contraction. And when that was finished, you had to put it into a solution of shellac, and now you had a permanent record. And I knew how to do that very well from Duquesne, but University of Illinois, they didn't have any of those. So Tech said, Dr. Stegerda, you need to study the manuals of the recorders. He was working in GI physiology. I, to this day, can't figure out why I chose to work with him. Probably because he wanted me to work with him. And he was working in those days on the effects of beans on flatus production. And I wasn't too interested in working on flatus production. So I was working on muscle contraction, and he was perfectly happy with that. He realized, though, that his level of research on muscle contraction was not equivalent to another one in the department, which was C. Ladd Prosser and Bud Abbott. And he said, if you work with me, you need to be aware of the cellular physiology, and Ladd Prosser is going to do that for you. So I had two advisors, Fred Stegerda and Ladd Prosser. Stegerda, at that time, just from a historical perspective, was the best funded investigator in the department. And that was because the USDA was very interested on Stegerda identifying the factor that created gas when humans ate a bean fiber. And the USDA was very interested in that because John Kennedy was president. He had a Food for Peace program, and they wanted to send soy flour to Eastern Asian nations. And the governments refused that because of the flatus production. True story. So they said to Stegerda, just tell us what you need, and you figure out what is the factor. And he did. It was a sugar called stachyose. And I can bring it forward right up to 2014 because stachyose, as we now know, is dependent on a microbiota. Okay, so you know, here we are 50 years later, and these two fields merging today in a very different but a very equally important way. So that was Stegerda. Prosser, entirely different faculty advisor, but he taught me quantitative physiology.
You did something that was probably unique for people getting their PhDs at that point in time. You didn't just go and maybe do one postdoc, but you actually went through 3 labs of the leading figures in physiology, smooth muscle physiology at the time, Charlie Code, Molly Holman, and Edith Bernstock.
Bullbring.
Bullbring.
Okay.
Sorry.
That's all right.
But tell us a little bit about how you happened to decide to make that journey.
Right. Well, just as a general comment, there were my colleagues who graduated with me from the University of Illinois when they found I was taking a second postdoc felt that he can't get a job. And so he's just delaying the fateful day when he has to recognize he can't be a scientist. So that kind of was a general comment, and I was getting back. Well, I went to Mayo Clinic because Fred Stegerda went there every summer to do research with Charlie Code. And I went there because Lad Prosser knew Charlie Code. And so my both advisors told me, you go to Mayo Clinic to do a postdoctoral research fellow. In those days, research fellows did what they were told to do. All right. So I was told, you go to the Mayo Clinic, and I did. And I worked with Charlie Code. About a year and a half into that experience, he said, we were writing a review chapter, and he went to the board, and I remember this vividly. It was almost midnight, and he drew a graph. And on the abscissa, he had effort, and on the ordinate, he had return. And if I were to draw the graph, it would be this way. And he said, Joe, you are on the asymptote of return, but your effort continues to increase and you're not advancing the field. So you need to go and learn a new way of doing research. Okay? Where do I go? He said, you go to England. And I said, fine. And he wanted that because he himself had gone to University College London, worked with Sir Henry Dale and a few other luminaries. He said, I want you to go there. Well, I applied for a Byrne Fellowship at Oxford. I was successful. A week after I found that I had gotten it, I got a telegram, in those days, telegrams, and it said, sorry, you can't come. You have to delay it by a year because the person who has it would like to stay for an additional year. So come a year later. On a whim, I decided to apply for a Fulbright scholarship to work with a student, Molly Holman, who had trained with Edith Pulbring. And I was interviewed by, of all people, Horace Davenport. So I drove up to University of Michigan, went into his lab. Remember that one as well because he had no secretary. He did all his typing himself. There was a sign on his door that says, do not disturb under any circumstance. So I waited for 30 minutes, finally knocked on the door, and he said, who is it? And I told him, and he said, can't you read the damn sign?
Sounded like Horace.
You know, and I said, and he was a friend of Charlie, and I said, well, I'm here for, oh, well, he said, you're late, you know. So got through that, went to Australia for a year, was going to work on muscle. Molly Holman said, no, I want you to work on peripheral neurons. Did a year there, picked up my fellowship at Oxford for 2 years with Edith Pulbring, and got a telegram from the Mayo Medical School saying that we would like you to come back just for a year to get your feet on the ground. You've been out of the country for 3 years. You have a family. You need to kind of figure out where you want to work. Well, turned out that's where I ended up working. So, yeah, three postdocs. And if I were to advise students today, I would say get as much training as you can, and by all means go out of the country to do it. Because you get a very different perspective of your own country, your own citizens, and you'll come back better informed. And I tell every person who finishes a postdoc with me, this is where you need to go, almost like Charlie Code. They don't listen, but it's okay.
Nor do our children.
Okay.
You clearly had phenomenal mentors. And how did those three experiences help shape the research program that you have pursued for the many years that you've been at Mayo?
It shaped it from the two perspectives. It taught me new techniques. And that was Charlie Code's warning to me. Don't beat a technique to death. Don't look for questions to ask using a technique. Go ask questions that you need to develop new techniques to answer. So I went off to Monash University in Australia and learned a new technique, new at that time, recording from single sympathetic neurons. And Molly's attitude was, just go and do it. If you have a question, find out how to answer it. I went to Oxford, same perspective, but in a more restrictive way. The British social scene is very different from the Australian scene. And of course, Charlie taught me, learn new methods, learn new methods, learn new methods, because that's the way your career will advance, and that's the way science will advance. So I suppose, Marty, the key issue was learning new approaches in a new environment.
In your long career, over 250 research papers and countless reviews and stuff, you've made some significant contributions to our understanding of smooth muscle motility and the role of the autonomic nervous system. Tell us a little bit about some of the high points you think of your career in terms of research contributions. And don't be modest.
Okay. All right. When I got to Mayo Clinic, we had the ability of suturing 40 permanent electrodes throughout the gastrointestinal tract and record from conscious animals. And what I observed, what has since then been called a migrating motor complex, which was a whole new discovery. And I take a lot of pride in that. And also, it was a teaching moment, because Charlie recognized the importance of that more than I did. And when I wrote the first draft for submitting to AJP, I got it back from him with his comments, but his name erased. And he said, "This is your discovery. This is going to be important for your career." That was a teaching moment. You know, advisors tend to cast shadows. He didn't. And just before coming here, I had a email from a patient who was discussing huge hunger contractions that this person had between meals. It was sent to Michael Camilleri, and Michael answered it and said, "What you're experiencing are the migrating motor complexes that Joe Szurszewski described in 1969. And since then, Marty, that complex has been found in virtually every animal it's been looked at, including birds, lizards, and it plays a key element in the irritable bowel syndrome. So I take a lot of pride in that discovery. The second one was in Australia, where we described for the first time what's called intestinal fugal neurons. And these are the neurons that connect the lower regions of the GI tract with the upper regions outside of the central nervous system. So they create a peripheral reflex loop. That was very controversial. Had a hard time getting it accepted, because no one wanted to accept the fact that there were neurons in the bowel wall that projected to peripheral abdominal ganglia, and that it used acetylcholine as a transmitter. And they absolutely refused to accept that, but we persisted and we got it accepted. The third one unquestionably reflects the environment I worked in, and that's Mayo Clinic, and that's with a team of surgeons and gastroenterologists. And I think my colleagues have the greatest collection of recordings from human biopsy material, bar none. And that to me is very pleasing. We have more data on normal human GI smooth muscle and diseased human smooth muscle than just about anywhere else in the world. So I take great pride in what my colleagues have given me. My career would never be what it is today without my colleagues in medicine and surgery. And so as part of that, we recorded from interstitial cells of Cajal, which are now, we know, the pacemaker cells of the GI tract. It's like the Purkinje system of the GI tract. That was an interesting and controversial one, because it contradicted Ladd-Prosser.
Right.
And when I made these recordings, I called up Ladd-Prosser and said, "Ladd, I'm going to send you a manuscript in draft form. I'd like you to look at it. Would you tell me what it is?" And so he called me back and he said, "Do you realize that you're contradicting my life's work?" I said, "That's why I sent it to you. I want you to tell me where I've gone wrong." So we worked it back and forth. He said, "You do it, you're right," et cetera, et cetera. So recording from interstitial cells for me was really a good area. The fourth one is the biological gases. Nitric oxide, carbon monoxide, hydrogen sulfide, and to cut to the chase, we found recording from material from patients who had emptying disorders that were diabetic that carbon monoxide actually can be used as a treatment to reverse diabetic gastroparesis.
Interesting.
So 4 areas I'm really happy with.
Right.
I would add, though, that as enthusiastic as I seem to you and as I describe it, I really reflect the work of everybody else.
Well, as we all say, we build on the shoulders of giants, and everything you have done so far will contribute to what somebody else draws upon for the future.
Right,
So,
and I didn't do it alone.
none of us can do it
I had
alone.
a tremendous cadre of postdocs, residents, and students and fellows, and I wouldn't be sitting here today telling you all this if they hadn't done so much work. So I'm grateful to them.
A couple of years ago, you and I were sitting in outside of a tent in Oxford, England,
That's right.
when you told me of a vision you had of creating a manual on motility called MOM.
Right.
And I know it hasn't progressed much further than having that conversation, but tell us a little bit about your ideas, and maybe by doing so, we'll find a publisher.
Well, thank you very much. I have found in teaching medical school students, graduate students, in my travels, that not many people pay attention to gastrointestinal motility. And in particular, the nursing staff. I found that when we interact with the nursing staff, they know they're going to measure esophageal motility or gastric emptying, et cetera, but the fundamental understanding of it is not always there. So I always felt that there had to be a simple textbook that described gastrointestinal motility. And if a paramedic technician came to work, someone euphemistically could say, well, have you read MOM, the manual on motility on this particular region? That might be useful. So I've always had it in the back of my mind to do that. But since I spoke with you, wrote a large chapter for the handbook, and I wrote 3 very comprehensive reviews on different topics, and that really, because I tend to want to make sure I get it right and keep it exactly up to date, has really occupied my time. I have a large program project grant, progress reports, competitive renewals. So MOM had a secondary interest because these were more primary. But today, if I were to write it, I will write it, I wouldn't call it MOM. I would focus more on rhythms and cycles. And I probably will call the textbook Rhythms and Cycles in the GI tract. So I'm
The acronym doesn't sound as good as MOM, though.
No, it doesn't. So you're convincing me maybe I need to force it into MOM.
Okay, sounds good. Speaking about MOM, obviously you mentioned your wife. You've been married for over 50 years.
Yes.
I think I would be remiss, and you may be remiss by not saying anything about her
Well,
and your family.
Cecilia and I, married for coming on to 51 years this August. I would not, I know one hears this, looks at books that are dedicated to wives, et cetera, but I would not have a career that I have without Cecilia. And it goes back to square one. We went to University of Illinois together when we just got married. She worked in Andy Nell Bandolph's laboratory.
Oh, yes, I remember Andy.
In genetics.
Yeah.
And she typed my thesis. She encouraged me. She really was there whenever I needed help and support. She did it. Typing a thesis that met the University of Illinois standards for whatever. It was not an easy thing to do, but she did it.
And I don't know about you, I've gone back and looked at mine, and there are a heck of a lot of typographical errors in there. So, and a lot of whiteout.
Yeah. Well, I went through mine not too long ago for a different reason, and I have to say I was hard pressed to find the typo. She is a perfectionist. Well, we have 2 children, one born at Carl Clinic, and another one up in Rochester. And so we have 2 children. Our daughter lives outside of Boston with 2 grandchildren. Our son lives in Minneapolis, not that far away, and we're just very, very proud of what they've accomplished. but Cecilia was the glue that kept the family together. So thank you, Cecilia.
Very nice. You've been around long enough to have experienced APS meetings on the boardwalk of Atlantic City. Tell us a little bit about your first APS meeting and perhaps how those meetings differ from the current meeings set.
All right. I knew you were going to ask me that because you said you would. So I thought back to that first meeting, and that was the meeting where I met Charlie Code. I was in Lad Prosser's office. He called Charlie and said, I want to send a student to you. His name is Joe Szurszewski. And at the other end, I guess, Charlie said, would you spell that? And so Lad started out S-Z, and he said, he's here, let him spell it for you. So I was told on the phone, I'm going to be in Atlantic City. You be there, I want to meet you. And so I met him at my first Physiology Society meeting on a windswept boardwalk. And those meetings were small, intimate, personable. There were no posters. I can't remember a exhibit hall where industrial...
That was in the old convention, that was in the convention center building where they would have the exhibits set up.
And I don't remember that. Probably didn't go to it. But it was a small, intimate meeting, and in those days, gastrointestinal motility was called miscellaneous science. Okay, fair enough.
We refined it a little bit better today.
You refined it better. So I fast forward to today, and my goodness, hundreds of people going back and forth from room to room, badges flying in the air, holding programs, standing on the elevator, learning about this and that. In fact, if I wanted to really bring myself up to date about what's going on, I think I'd ride the elevator all day in the Marriott Hotel. But what impresses me is the enthusiasm and the passion that the young people have for what they're doing. And I would say that, and I say that to my students, you've got to have passion for what you do. Do not ever call it a job. Do not ever say you're going to work. I can't think of anything worse, Marty, than going to work in the morning to do a job. Okay? Because we don't do it 8 to 5, do we? We do it all the time now, at night, on weekends. And I can't think of a job I would want. I can't think of a work I would want to do that I did 7 days a week. So it's different today, and it's exciting.
Sounds good. Since you started talking about the enthusiasm for stuff, for the research and all full-day job, so to speak, give some of the young people additional advice on how to shape their own careers.
Well, students tend to think they have to go to a named university. When I was talking with, and I'll mention his name, Malcolm Moose, President, University of Minnesota. And I'll never forget him saying when our children, or Joseph and Marie, our children, were thinking of going off to university. And he said to me, what's very important is wherever your kids go, they want to go to. The name is not that important. Because if you make them go to a named place and they never wanted to go there, they will not do as well learning-wise. And then if they go to a place they really wanted to. So my first comment would be, go where you really, really want to learn. Don't listen to your parents, okay, number one. Number two, surround yourself with excellence. Surround yourself with people who are better than you. Because if you do that, you'll realize you're not as good as they are, and therefore you will try to be as good as they are. And when you become as good as they are, move on to be in an environment where there still are people better than you. And thirdly, as I already mentioned, have a passion for what you do. Don't worry, we may get into this or not, about the extramural NIH funding scenario. We can get into that if you want to, but if you believe in what you do, if you surround yourself with excellence, it'll work out. That's what I tell them.
Well, I want to keep this upbeat, so I'm not going to talk about NIH.
Okay.
You've been involved with societies, you've been a member of APS for a long time. You've never risen to an officer role in APS, but you have in other societies. And talk a little bit both for your own career and even for students, the importance of a society to help shape an individual's career.
It's a calling card, isn't it? I hate to use the word union card because in this country today, the term union doesn't make people a lot of happy all the time. My involvement with societies has more been involved with a society, the American Gastroenterology Society. So you might wonder why, if you're a physiologist, and the answer is very simple. I worked in the medical institution and I benefited from the surgeons and clinicians who worked with me. And so it was only natural, if I were studying human tissue, that I would present my work at a society that was concerned with the human condition. But I never forgot I was a physiologist. And when I joined the Mayo Clinic staff, I was offered a laboratory to work with Bill Summerskill, who was a giant in liver physiology, in St. Mary's Hospital. And I refused to go there, and he said, "Why would you do that?" And I said, "Because the physiologists are in the medical sciences building, and my union card says physiologist." And so I never forgot Physiology Society. I was always a member since 1973, came to virtually all of its meetings, but I was more akin to and related to the clinical side. Today, if I had to start all over as I did then, I probably wouldn't feel so out of left field in physiology today, because it's translational, isn't it?
Right.
That's what we do in this society. So today, I probably would have tried to have office in the Physiology Society, but I didn't back then. And I did rise to offices in the AGA, American Gastroenterology. So.
You raised an interesting issue, and it's in the role of physiology in translational medicine. And obviously, if you listen to, I'm sorry for bringing this up, NIH, one of the areas they seem to be pushing is translational research, translational medicine. How do we fit in?
Well, you're talking to a converted person. I feel I've done translational work my entire career. And I say that because I used human biopsy material. So for me, when the NIH decided, what was it, 15, 17 years ago, to emphasize translational research, I'd say, okay, fine, you know, I have no problem with that. Science changes with time. And I remember we went through an era in this country where molecular biology, cell and molecular biology, was the thing to do. And the physiology departments were falling over themselves to change their name from the Department of Physiology to the Department of Cell and Molecular Biology. And I was chair of physiology at Mayo at that time. I thought, this is ridiculous. You know, physiology is function, structure. You can't get more basic than that. And what molecular and cellular science does is to fill in the physiological importance of what it is that one's studying. So I say the NIH, well, yeah, physiologists by definition do translational research. You can't predict that what's fashionable today is going to retain its fashionability 10 years from now. So please don't restrict us, and please look at our science as a critical field. I'm kind of repeating myself, but it's the best I could do with that one.
Well, I appreciate your comments, and I want to thank you for being part of the Society's Living History program. I'm curious if there are other things you might like to talk about from your own career that I may have missed.
No, I think you know me, so you know pretty much my career.
Well, I am pleased to do this interview, and I'm really pleased to see you still have the same passion and enthusiasm for science, and that's very gratifying for me.
Well, it's because we have societies such as you're the executive secretary of that keep it alive.
Well, thank you, Joe.
Yeah.
Thank you for the opportunity. I'm sure lots of people will watch us, and hopefully a few people will get in touch with you and ask you a little bit more about your
Great.
career.
Thanks,
Thank
Marty,
you.
for asking me.